This is Dr. Byson AM. Today we're looking at Histology Practical 2, which is the first histology lab that you have. Make sure to subscribe to this channel and like and comment. So the histology lab that we're doing today is the cell and cellular organelles. All right, so let's get into this lab and see what is expected for us to do and to write. Okay, the introduction. So in this experiment, we're using a microscope to study cells and cell organelles. So remember this, we are studying cells and cell organelles in this experiment. That is our interest. Okay, so the cells have been stained and have been captured from a microscopic slide, and from that slide we're drawing the cell and mentioning the parts and the organelles that are found inside of a cell. So the microscope is an instrument that is used to study objects that are too small to be seen by the naked eye, and the cells cannot be seen by the naked eye. Therefore, a microscope is used in the study of cells. This is the branch of anatomy that is called cytology. So you're using the microscope to study your cells. That is called cytology. So for your cells to be visible in the microscope, they need to be stained. So we use histological stains. Histological stains are simply biological material that is inherently will allow you to study the parts of the cells, because they will go and stain and highlight organelles in the cells. As we go on in this video, we're going to see the type of stains that are used in the study of cells and tissues under a microscope. So hematoxylin and eosin, which is just commonly abbreviated as H&E, are the most widely used dyes in histology and in pathology. All right. Now, when it comes to dyes, there are two types of dyes. There are basic dyes and there are acidic dyes. So we use a basic dye and an acidic dye that stain tissue components selectively. So tissue components that stain with basic dyes are termed as basophilic, and they stain blue in color. So when you look at them in the cells, they appear blue or purple. And tissue components with an affinity for acidic dyes are termed acidophilic, and they stain pink or orange in color. So always remember this. This is very, very important. Now, hematoxylin and eosin staining, as already mentioned, is the most widely used method for preparing slides. So basophilic structures such as the nucleus stain dark blue or purple by this method, whereas acidophilic materials such as collagen stain, they stain pink or orange. Okay. So H&E staining can be used successfully to study a wide variety of tissue types and organs. So it's very widely used. All right. Now, what are the learning objectives of this lab? At the end of this lesson, the students should be able to, number one, define what the cell is, to describe the general features of the cell, the shapes and the size of cells, to describe the function and the structure of the cell membranes, to distinguish and describe various cellular organelles as seen under both the light and electron microscopes, to differentiate cytoplasmic organelles from inclusions, to describe the structures of chromosomes and the karyotype of a man, and lastly, to describe and explain the processes of mitosis and meiosis. Okay. What is the goal of our experiment? The goal of this experiment is to learn how to look for items of interest in histological specimens using the light compound microscope and to identify them, starting with the cell. So today we're looking at the cells. Okay. So the characteristics that we're going to notice in this lab and to observe: number one, we're going to observe the shape of the cell. Secondly, the size of the cell, the nuclear and the cytoplasmic ratio, the size of the nucleus in relation to the size of the cytoplasm. Fourth, the chromatin condensation. So we're going to look at heterochromatin and euchromatin. And we're going to look at cytoplasmic staining, whether it is basophilic, which is blue or purple, or acidophilic, if it is staining red or pink. Okay. Now let's define what cells are. These are just basic and functional units of all living organisms. And these cells vary in size and in shape depending on their functions. And microscopes are the devices or the instruments that are used to study these cells. So all the slides that I'm going to project in this presentation will just be looking at cells in different tissues. Yeah. Now let's start looking at our slides. The slide one that we're looking at are slides of sebaceous glands. Okay, sebaceous glands. So now, in the next slide, you have to look for sebaceous glands. Sebaceous glands will appear as clusters of pale cells with distinct central nucleus. So your task when you look at this sebaceous gland is to draw each cluster at low power or high power, and then draw it at high power. So at high power, you're supposed to draw 4 or 5 adjacent cells that are together. And then you label the plasma membrane, you label the nucleus, you label the nucleolus, and the cytoplasm. Okay, so you make 2 drawings, one at low power or medium power, and the other one at high power. And then you label all those organelles. So this is a slide. So this is the sebaceous gland. So sebaceous glands are found in thin skin. Okay. Thin skin. So this slide was taken from the scalp. The scalp is the skin that covers your scalp. Okay. So this sebaceous gland, as you can see, is turning very pale. So this area here is very pale. And these dark things here are what we call nuclei. So this is a cell with its nucleus. This is a cell with its nucleus. This is a cell with its nucleus. So you see a lot of distinct nuclei. The sebaceous gland contains a lot of cells. Okay. So this is a sebaceous gland at very, very high power. At high power. So at high power, this is the cell. So this is the cell, and what is inside here is the nucleus. Inside of the nucleus, this dark stain here is the nucleolus. So you're supposed to draw this cell. So draw this cell and this cell, these 2 cells. And then label, this is the plasma membrane, this is the cytoplasm, this is the nucleus, and this is the nucleolus. That's how you're supposed to name and to label them. So that is it about the sebaceous glands. So this is a slide at low power. You draw this at low power, and this is a slide at high power. So make sure you make a drawing at low power, and you also make a drawing at high power. Okay, let's move on. Slide 2 is the sensory ganglion. So on the sensory ganglion, you're going to examine the large grayish nerve cell in the section that I'm going to show very soon. And you're going to note the presence of a large nucleus in the cell with a prominent nucleolus and cramped chromatin. The cytoplasm contains a network of fine fibrils, which are also known as neurofibrils, as well as dense granules representing the rough endoplasmic reticulum, which are called Nisi substance. Each nerve cell is surrounded by numerous small, darkly stained nucleus of cell called satellite cells, and the wavy lines around the groups of the nerve cell are called nerve fibers. So all of this information I'm going to illustrate in the slide that is coming. So the task here is to draw and label 3 or 4 nerve cells with their satellite cells. Okay. This is what we're talking about. So this here is a nerve cell. Okay. So this is a nerve cell. The outermost part is the plasma membrane. This whitish part is the cytoplasm. This that is dark here is the nucleus. And this central part here that is a little bit light is the nucleolus. Now, you notice that, okay, so you notice that this cell is surrounded by this nucleus here that's stained dark. This one here, this one here, this one here, this one here, and this one here. So these dark nuclei that are surrounding the cells are what we call satellite cells. They are called satellite cells. So when you draw these cells, make sure you include these inclusions. Okay. The satellite cells, because that's what we want to see, and they carry marks. These wavy-like substances here, these are what we call axons. Okay. They are axons of the cell bodies. So this is a cell body, and this is an axon. So make sure you draw 1, 2, 3 cells. You label their parts, and you also include the satellite cells. So that's it about the ganglion. So this is just a magnified picture of what I've been showing you. Okay, so this is the cell, the neuron. This is the cell membrane, the cytoplasm, the nucleus, the nucleolus. Inside of the nucleolus here, that's where you find chromatin. This is chromatin here. And then this dark nucleus that are surrounding this cell membrane are what we call satellite cells. Okay. So this is also a motor neuron. Okay. So as you can see, this is a multipolar neuron because it has a lot of dendrites. So it has this dendrite here, this dendrite here, this dendrite here, this dendrite there. Okay. So inside of the cytoplasm, you also find Nisi granules. This is a nucleus here, and then this is a nucleolus. The outermost part is the cell membrane. Okay, let's move on to slide 3, which is the liver. To stain the liver, we use a stain called the periodic acid skiff, which is denoted as PAS. So this tissue was fixed in a special alcoholic fixative that is called camel in order to preserve the glycogen stored inside of the liver cells. Remember, the function of the liver is to store glucose in form of glycogen. So when you are staining the liver, we use a special alcoholic fixative to preserve this glycogen that is stored in the liver, which now appears as intracellular red granules. They appear as intracellular red granules after staining with the periodic acid skiff. Our assignment is to draw and label a few cells at high power. So now let's look at this. So these are the liver cells. The liver cells are called hepatocytes. Okay. So this is low power. So this is the liver cell with its nucleus, liver cell, nucleus, liver cell, nucleus, liver cell here, its nucleus, liver cell here with its nucleus. At high power, it will look like this. So this is the liver cell here. How do I know? It has a nucleus here. This is another liver cell here. How do I know it has a nucleus here? Now, inside of the liver cells, there are these red granules here. These are glycogen granules. So, draw 2 or 3 liver cells. Label its plasma membrane, its cytoplasm. Inside of the cytoplasm, include the glycogen granules. So, show the glycogen granules and also mention the nucleus. Draw 3 to 4 liver cells. Okay. Now let's look at squamous epithelia. Squamous epithelia, remember when we were looking at epithelia, when you were looking at epithelial tissue, we said squamous epithelia, one of the regions where it is found is the esophagus, the mouth, or the vagina. It is also found on the skin, depending whether it has keratin or not. Okay, so the preparation that we're going to look at is a smear of epithelial cells from the cheek, which is the mouth, the buccal cavity. So you draw and label 3 or 4 single cells at high power. Okay. So this slide that was given in the lab is not very clear. Okay. But this covering here will give you epithelial cells. So since this slide is not clear, I'm going to show you this schematic diagram. This is how epithelial cells look. They are very flat cells. They are wider than they are long. They are very flat cells. So this is one epithelial cell, one epithelial cell. If you look at them in 2D, they appear like this. This is one epithelial cell here, one epithelial cell, one epithelial cell. Remember that epithelial cells are polyhedral cells that are tightly held together by tight junctions. A video that we have made, a link will be in the bio. Go and look at tight junctions. Okay, so here is a another sectioning. So this is an epithelial cell here. This is an epithelial cell. This is an epithelial cell. So draw one that is in this section and draw another one that is in this transverse section. So pick 4 here and draw, and then pick 3 here also in this transverse section and draw. Okay. So label the cell membrane, label the cytoplasm, label the nucleus. All right. So for epithelia, always also mention the basement membrane because the cells lie on the basement membrane. So always make sure to mention the basement membrane for epithelial cells. Okay, so our last one, let's look at columnar ciliated epithelia. Columnar epithelia mostly is found in the GIT cavity, and it's also found in the respiratory cavity. Mostly in the respiratory cavity, it is pseudostratified. Okay, so in this slide, the task is to draw 3 or 4 suitable cells, and you draw them at high power to show the cell membrane, the nucleus, and the cilia, which extends from the free surface of the cell. Let's look at the slide. So this is a slide. This is a simple columnar cell here. This is a simple columnar cell, simple columnar cell, simple columnar cells. So just pick 1, 2, 3, 4 and draw them. Okay. So label the cell cytoplasm, label the plasma membrane, the nucleus, and mention the basement membrane, because remember, epithelial cells always lie on the basement membrane. So this is simple columnar epithelia. How do I know? Because the nucleus are at the same level. So this is simple columnar epithelia. So that is what you're supposed to do in this lab when it comes to the drawings. So make sure on your plain paper, just make 2 drawings on one surface. Do not draw back to back, but just draw on one surface, and on each plain paper only 2 drawings. Right, so now the exercise that is to be submitted for marking together with the diagrams is this. Number one, with well-labeled diagrams, compare and contrast prokaryotic and eukaryotic cells. Prokaryotic and eukaryotic cells, compare and contrast by drawing as well. Secondly, living organisms are divided into how many kingdoms? Answer this and state the kingdoms and the type of cells which make up these organisms. 3, you should differentiate cytoplasmic organelles from inclusions. What is the difference? And mention some of the examples that we've mentioned in this lab. 4, with a well-labeled diagram, explain why a cell membrane has a phospholipid bilayer. Explain or show the phospholipid bilayer of a cell membrane. 5, what is the function of lysosomes? List any 5 lysosomal storage disorders. This is a very important question because it also comes as an essay in histology. So make sure you answer this one and remember it. 6, briefly explain the hematoxylin and eosin, silver staining, Nisi staining, and periodic acid skiff technique. 7, colorate the structure of the mitochondria, the nucleus, the smooth endoplasmic reticulum, rough endoplasmic reticulum, and Golgi apparatus to their functions. So know the structure and their functions. 8, describe the structures of chromosomes and the karyotype of a man. 9, with label diagrams, describe and explain the phases of mitosis and meiosis. Number 10, you should explain the steps of how information in DNA finally gets translated into proteins. So here, it's like translation. You just explain from transcription to translation. This is what you did in molecular biology last year. So this should be very easy. Right. So in your private study, you should use this material. Study the diagram showing the general structure of the cell as reviewed by the electron microscope. So you're not supposed to draw structures from an electron microscope. Draw structures that you have seen in this lab, the slides that I have shown you in this lab. Okay, but in your private time and in your group discussions, use the electron microscope to study in detail the structure of the cell. Okay, there are certain inclusions in the cell that we cannot see with a light compound microscope, but the electron microscope will show you. For your private study, use Janquilla, which is a textbook of histology, chapter 2 and chapter 3. Then you will fully understand the cell and its organelles. All right, I hope this video was very helpful, and if you were lost in any way, I hope this video has brought liberation to you. If any questions, ask in the comment box and I will gladly respond. Thank you. And if you need lessons, you can register at our academy on this number 0975497790 and let us learn together. Thank you very much, and I'll see you in the next video.